Welcome into the second part of this video, which is process of RDT, that means recombinant DNA technology. In our previous video, we studied about the processes of recombinant DNA technology of three processes, that means first, second and third. And in this video, we are going to study the next three process of RDT, that is fourth, fifth and sixth. That means amplification of gene of interest using PCR, fifth, ligation of the DNA fragment into a vector, sixth, transferring the recombinant DNA into the host. So these three processes we are going to study in this video. If you want to know all these steps, you have to watch this video till end. And in this video, PCR is an important topic with respect to the exams. If you want to know all the points of PCR, you have to watch this video carefully. So stay tuned, and if you are new to our channel, please subscribe to our channel, will be amazing. And the part 1 of the video, or say previous videos, link is given in the description. If you haven't watched that video, you can go and watch that first and then you come into the second video. So the fourth number is amplification of gene of interest using PCR. It means, as we know, we can extract a little amount of DNA from a bacteria, because bacteria are already very small and the DNA present inside the bacteria are also little. So we have to amplify the DNA extracted from that bacteria. For this purpose, we use PCR. PCR is a technique to replication of DNA to huge amount in a minutes or in a hours. So PCR is discovered in 1970s. During that time, for one cycle in the PCR, the enzyme which is used, polymerase, DNA polymerase, is required for every cycle, because in PCR high temperature is required so that the DNA polymerase always denatured due to high temperature. So always in new cycle, new DNA polymerase required, so that process becomes tedious and errors. So for this purpose, we are going to study all the components of PCR which are currently using nowadays. So first is the most important, that is Taq polymerase. Taq polymerase is also a DNA polymerase which is extracted from Thermus aquaticus bacteria, which lives in high temperature areas, and this Taq polymerase has a property to survive at 95 degrees Celsius. At 95 degrees Celsius, this enzyme do not denature, and this is useful for PCR, because in PCR high temperature is required for the amplification of DNA. Second component of the PCR is initial template. For the formation of new DNA, template DNA is required, and this template DNA is taken in a small amount, or we can say that taken in nanograms. The third component is the substrate. For the formation of new strands of DNA, substrates are required, which are nucleotides, or 4 substrates are required for the formation of new DNA, that is deoxyribonucleotides. This helps to make the new strands of the DNA. The fourth component of the polymerase chain reaction is the primers, which is an important component of PCR. Primers are synthesized oligonucleotides which binds to its complementary DNA. Without primers, PCR is not possible, because Taq polymerase has the ability to polymerize the replication of DNA but not has the ability to start the replication. That's why primers is required for the initiation of replication. That's why it is an important component. And the fifth component are few necessary ions and salts. Now we have discussed the components of PCR. Now it's time to study the steps involved in PCR, which we call cycle. That means there are 3 steps involved, which are temperature dependent steps, and these 3 temperature dependent steps form a complete cycle of PCR. So the first step of the PCR is denaturation. That means denaturation of DNA molecule, in which the mixture which contains the DNA molecules is heated to 94 degrees Celsius for a couple of minutes, because as we know, Tech polymerase can survive up to 95 degrees Celsius. So we heat this solution which contains Tech polymerase, primers and DNA molecules to 94 degrees Celsius, so that the hydrogen bonds present between the two strands of the DNA broke down and they separate from each other. And now the second step comes is the annealing. In this step, the solution temperature is bring down in between the 50 to 60 degrees Celsius for a less than a minute. That's enough for the primers to bind to its complementary strand. That means in this step, primers bind to its complementary DNA molecules. As you can see in the diagram, in first step, DNA strand separates, and in the second step, primers bind to its complementary DNA. As we know, primers are exposed 3' end, so the Tech polymerase can only polymerize from 3' end of the primers. And now the third step comes is the primer extension. That means polymerization. In this step, DNA polymerase, or we can say that Tech polymerase, act on the complementary primers or molecules and start replication. This step occurs at the 72 degrees Celsius, and these 3 steps combinedly form a one cycle of PCR, and if we do 30 cycles of the PCR, 1 billion copies of DNA molecule can be formed. So the formula which comes of PCR is, after n cycles of the PCR, to the power n molecules are formed. And now the fifth step comes is the ligation of DNA fragment into vectors. For this purpose, we already know how the restriction enzyme works on the vectors and on the DNA. For this purpose, same restriction enzyme used. You can watch the activity of restriction enzyme, that how it works, at the I button which is showing and in the description link. Now the sixth step comes is the insertion of recombinant DNA into the host cell or organism. In this process, we have already made a video which is named as competent host, which tells how the host organism is made competent, in which all these steps are already explained on that video. Now we are going to again explain these in little way. That is, first step is transformation, in which bacterial cell is treated with divalent cation, that is calcium or say calcium chloride treatment, so that the cell wall of the bacteria take up the plasmid, and then with the help of heat shock, plasmid is inserted into the bacteria. The second step comes is the microinjection, in which we directly inject the DNA into the nucleus of the cell. And the third is biologistic or gene gun, which is used for plant cells, in which heavy particles are bombarded on the cells which are coated with the DNA molecules. Now in this video we have already discussed the 3 steps of the RDT, that means recombinant DNA technology. If you like this video, subscribe to our channel, will be amazing. Thanks for watching. If you are watching this video after a few days, then maybe the third part of this video is uploaded to here. Watch this